you hear me now? Check, check. Testing 1, 2, 3. Testing 1, 2, 3. Check, check. Test.
Okay, welcome everyone. I'm going to keep the introduction super brief so we can have more time to hear Liza Comita. It's a really great pleasure to introduce Liza. She is a tropical forest ecologist here at the school, for those of you who don't know. She is incredibly prolific. She has over 60 scientific publications. She's been cited over 5,000 times. She's won all sorts of awards, including the Founders Prize for Early Career, which is given by the British Ecological Society. She is an Early Career Fellow of the U.S. Ecological Society, or Ecological Society for America. On campus, it was either last year or the year before last, she won a major prize for mentorship. I see Liza on all sorts of lists in terms of mentorship of postdocs, mentorship for students. So she is not only an amazing scientist, she does a lot in terms of mentoring our emerging scholars. So I'm just going to keep it brief so we can hear Liza for the next 49 minutes. So please join me in welcoming Liza. Thanks.
Thank you. All right, is this on? Okay, great. So thanks, Karen, for the introduction, and thanks to all of you for coming out. I'm really excited to be telling you about work we've been doing in my lab over the last several years focused on species interactions and how they contribute to the maintenance of diversity in tropical forests. So as everyone here probably knows, tropical forests are very well known for having extremely high diversity. They have the highest terrestrial diversity on the planet. And within a single tropical forest site, in the most diverse tropical forest, you can get up to hundreds of different tree species coexisting in the area the size of a couple football fields. And this is compared to, in all of continental Europe, there's only 134 different tree species. In an area the size of a couple football fields, in the tropical forest, you can have even more. And so this is an amazing number of species. And it also poses a challenge to ecologists to explain how can you have so many of these species coexisting? And the reason this is a challenge is because of an idea in ecology called the competitive exclusion principle. And the idea is that if all species are competing for the same limiting resources, if any one of those species is just a hair better competitor, it will push the other species to local extinction. And if you're talking about animals, it's not that hard to explain how species avoid competitive exclusion. This bird eats insects, this one eats big fruit, this one eats little seeds. But when it comes to trees, it becomes a lot harder because all plants depend on the same few limiting resources: light, water, and a handful of soil nutrients. And so how on earth can you get 200 different tree species growing in the area size of a couple football fields? And so this has long plagued ecologists. And they've been thinking about it for a long time. We've been thinking about it. And there's no shortage of hypotheses that have been proposed to explain how you get this extremely high diversity maintained in tropical forests. And I'm not going to go through all of these different diversity hypotheses, but what I want to emphasize is that we start talking about how you explain all the diversity you see here. What scientists have focused in on is not what's happening with these larger adult trees, but what's going on the forest floor. What is driving spatial and temporal patterns of recruitment in the forest understory? And those processes are really what's key for explaining how you get so many different tree species growing together. And in my lab, we focus on these early life stages on something I like to think of as the regeneration pathway. So you have seed dispersal. You have these seeds dispersing away from their parent trees, creating this spatial pattern on the landscape of seed fall. And then you have interactions between intrinsic traits of species and their biotic and abiotic environment. And so what I mean by that, in terms of the biotic environment, things like seed predators, insect herbivores, mycorrhizal fungi that interact with these seedlings and seeds. On the abiotic side, we have things like light availability, water availability, temperature, soil nutrients. And these interactions are ultimately what shape the patterns of plant recruitment that allow for species to coexist. In my lab, we focus on this regeneration pathway, in particular on these interactions at the early life stages between the biotic and abiotic environment. A lot of my work focuses on the abiotic environment in terms of drought impacts, but for the sake of time today, I'm going to focus on the work we've been doing looking at the role of the biotic environment and how that affects patterns of plant recruitment and how that can maintain diversity. But before I go any further, I want to explain why this is important. So you might think of tropical forests as these pristine, remote systems occurring, the heart of the Amazon. But like every other ecosystem, tropical forests occur in a changing world. So throughout tropics, you have alarming rates of deforestation that leave remaining areas fragmented and opened up to other human impacts. Like hunting, with poaching occurring at scales that's often not sustainable. And like every other place on the planet, tropical forests are experiencing and are predicted to experience increases in temperature, as well as shifts in rainfall, both in terms of the amount of rainfall and the timing of that rainfall. And there's a recent study that came out a couple years ago where they looked at the impacts on biodiversity of these human disturbances that are happening in intact, what appears to be intact forests. Things like hunting, selective logging, forest fires, and livestock encroachment that come into these forests. And they were working in this system in Brazil, and what they did was they calculated the combined effects of these various human-driven disturbances on biodiversity losses. And what they found were that these human disturbances were causing biodiversity losses on a scale similar to or greater than those caused by current deforestation alone. So certainly when you go through and you cut down a forest, you lose massive amounts of biodiversity. But for all the forest that was remaining, they were experiencing these disturbances, and that was also causing massive biodiversity losses. And that links back to an idea that's been around for a while in ecology, this idea of the empty forest. And there was this classic paper back in 1992 where Redford wrote, "We must not let a forest full of trees fool us into believing that all is well. An empty forest is a doomed forest." And for that paper in particular, he was focused on the loss of large animals, big mammals, large birds. But this holds for any kind of ecological interaction that's important for maintaining ecosystem function and biodiversity. So things like interactions with seed dispersers or with microbial pathogens or microbial mutualists. If we are losing those interactions, what looks to be an intact forest is actually a doomed forest. So in my lab, what we focus on are asking questions about what are the key ecological interactions that maintain diversity in tropical forest communities. And this is really questions that relate to fundamental ecology. But we also talk about questions that are more applied. Things like, well, how are anthropogenic disturbances in tropical forests altering these interactions? And finally, I'm going to talk to you about some work I have planned for the future, looking at whether we can recover these diversifying interactions over time as forests regenerate. And so as I mentioned before, today I'm going to focus on biotic interactions. And I'm going to focus on a class of biotic interactions that cause something called conspecific negative density dependence. By the end of this talk, you are going to be sick of hearing that phrase. But if you want to understand anything I'm about to talk about after this, you need to understand what this is. And it's really a simple concept. It's the idea that an individual growing here is going to have a lower probability of survival if it's surrounded by neighbors of its own species than if it's surrounded by neighbors of other species. So why might you get that? Well, there are two potential mechanisms. One is that these two seedlings that are the same species, they probably rely on the same limiting resources, and so they're going to be competing really strongly. But a lot of evidence suggests that this is actually driven more often by host-specific natural enemies, things like these insect herbivores or fungal pathogens that attack plants of the same species. Okay, well, how does that maintain diversity? The idea that these natural enemies can maintain diversity has been around for a long time. It was formalized back in the 1970s by Jansen and Connell in this enemies hypothesis, and I'll walk you through it. You've got this reproductive sized tree here, and it disperses seeds, this line here, and most of those seeds fall close to the parent plant. So you get this high density of seeds and seedlings right near the parent plant. And what happens is this high density of seedlings of all of the same species close to the parent plant attracts their host specialist insect herbivores that like to eat them, or it facilitates the spread of pathogens throughout these individuals. I always imagine it like walking in to drop my kid off at daycare. You have this high density of young, susceptible individuals, and you see that one kid in the corner with the runny nose, by the end of the week your kid is going to be sick because you have this high density of susceptible individuals that are all susceptible to that same thing. And what happens in plants, which luckily does not happen in humans, is as a result, offspring have a very low chance of surviving close to their parents at these high densities. Again, luckily this does not happen in humans. And what we see is that the probability of a seed or seedling surviving is very low at these high density sites close to parent plants and increases as you move away from the parent plant. And so what ends up happening is you have this peak in recruitment probability at some distance away from a parent plant, where you still have seeds arriving, but they have a better chance of surviving. So how does this maintain diversity? Well, if you think about it, if this tree is the only tree of its species in the whole forest, and it manages to get some seeds out here, those seeds are going to survive. But if this is a species that's really common, when those seeds move over here, they're going to end up just bumping into other individuals of their same species. And what this means is that seedlings of species, when they become rare, so rare species, they're less likely to encounter their enemies. They can escape by dispersing away from their parent plant. But for common species, they can't escape. Anywhere they go in the landscape, they're going to bump into their host-specific natural enemies. And this creates an advantage for rare species that keeps them in the system, prevents them from being forced out and going locally extinct, and it creates a penalty for being common. So as you become more and more common, fewer and fewer of your seeds and seedlings can escape these natural enemies. And so in my lab a few years ago, we did a meta-analysis. So we did a comprehensive search of the literature and pulled out values. And we specifically looked at studies that were testing these distance and density dependent predictions of the Jansen-Connell hypothesis. And we limited it in this case to experimental studies. So people either had to manipulate the distance that a seed or seedling was from a large tree of its own species, or they had high and low density treatments. And this is showing the overall effect size across all of the 154 studies we found. And what we found was all of the effects are significantly negative for the most part. So being close to a large adult of your own species or occurring at higher densities means that you have a lower chance of surviving. This was particularly true for seedlings. It wasn't significant for seeds. What I'd also like to point out is this isn't just a tropical phenomenon. This is something that we also saw repeatedly in temperate plant communities. And so the dynamics I'm talking about today are relevant not only for tropical forests, but for temperate grasslands or temperate forests. And so there's good evidence that these types of dynamics are occurring. And for the last almost 2 decades, I've been looking at these types of interactions that are occurring specifically in one tropical forest on Barro Colorado Island, which is in central Panama. And BCI for short, Barro Colorado, BCI for short, is this island here, and it's actually a former hilltop. They dammed a river and flooded this area to form a part of the Panama Canal. I think they got tired of digging, and they realized they could flood it. And so what happened is this became an island that's isolated from the mainland, not by very much, by very far, and it was leased long-term to the Smithsonian Institute to be used as a biological research station. And this is lowland tropical moist forest. It gets about 2,600 millimeters of rain a year. there's also this pronounced 4-month dry season, and it's considered semi-deciduous tropical moist forest. And so we think of deciduous meaning trees around here that lose their leaves in the winter. In the tropics, at this site in particular and other dry sites in the tropics, you see trees that lose their leaves in the dry season. So Craig talked last week about when trees open their stomates to photosynthesize, they lose water. Well, in this case, they avoid that by actually dropping their leaves in the dry season. And it's not a bad place to work. There are some nice dynamic animals there, seed dispersers and birds, monkeys. It's a pretty fun place to work. And a lot of people work there. Hundreds of scientists come from all over the world to work on Barrow, Colorado Island every year. If you're a plant ecologist, though, the main reason you come is this rectangle right here. This is the Barrow, Colorado Island 50-hectare forest dynamics plot. And it's an area that's 1 kilometer by 1/2 kilometer right in the center of the island. And back in 1980, they set up this plot to monitor forest dynamics. And in this 1 kilometer by 1/2 kilometer area, they went through and they tagged every single shrub, sapling, and tree greater than 1 centimeter DBH. So DBH, for those of you not in forestry, is diameter at 1.3 meters above ground. And they went through and they tagged every single one of these stems. It ended up being nearly 250,000 stems. They gave them a tag, they measured them, and they mapped them, and they identified them to species, and they encountered just over 300 different tree and shrub species at this site. And they've gone back every 5 years and census these trees. So we have this very large, long-term dynamic data set. But what they realized after a while is a tree that's 1 centimeter in diameter actually can be extremely old. So a number of these species are very slow-growing, shade-tolerant species. They grow very, very slowly over time. And someone did a back-of-the-envelope calculation, and they came up with this median value. The median age of a 1 centimeter diameter sapling is actually 80 years old. That means we've been missing 8 decades in the lifetime of many of these individuals. And as I mentioned before, we think that the key mechanisms that are maintaining diversity in these forests are happening at these early life stages, and yet we're missing all of that information. So in 2001, I led the initiation of a seedling census in this 50-hectare plot. And what we did was we put a 1 by 1 meter square seedling plot in the center of every 5 by 5 meter section of this 50-hectare plot. If you do the math, that's 20,000 of these seedling plots, which were measured with a lot of people, a big team. And for every seedling in these plots that was at least 20 centimeters tall, we gave them one of these plastic poultry bands. They actually use them on poultry farms around chicken legs, and they expand as the tree grows so they don't choke anything, but they also stand up to the tropical climate. And each seedling that got a tag was measured for height, it was identified to species, and it was mapped. And in this initial census, we encountered about 61,000 different seedlings of just over 340 species of tree, shrub, and lianas, which are woody vines. And what we've done is we've gone back every 1 to 2 years and recensused these plots. So the most recent census in 2018 finished about a month ago, and I think we're just about at having 120,000 unique seedlings tagged through this project. And this is a great data set for asking these questions about the effects of density, of conspecific density. And so what we can do for every seedling with a tag, because this is a spatially explicit data set and we can link it to this other spatially explicit data set, we can say, okay, for this seedling growing here, how many other seedlings are growing around it in this plot? How many of those seedlings are of its same species versus a different species? And then we can look at the XY coordinate of the seedling and the XY coordinate of the trees to figure out who's growing around it and ask what's the density of adult trees of its own species versus adult trees of other species. And we can use that information to construct what I call a neighborhood model of seedling survival. So what we can do is we can model the probability of an individual seedling surviving as a function of the density and identity of its neighbors. And we can divide that up into the effect of seedling neighbors and the effect of adult neighbors, and we break that out into conspecifics, meaning same species, same species as the focal individual, versus other species, heterospecifics. And as I've mentioned before, based on this meta-analysis and other studies, we already know that from many studies that there is this local scale negative effect of conspecific neighbors, both in temperate and tropical plant communities. But what no one was really thinking about at the time was how much the strength of that effect might vary among different species. This is a system with 300 different tree species. So I'd be pretty surprised if we went out and we found that every single species was equally affected by this phenomenon. And so I asked the question, does the strength of conspecific negative density dependence vary among species within a community? And what I mean by that, by the strength, is the decrease in the probability of survival due to the addition of a conspecific neighbor. And so I'll just explain that with an example. If we have species A here, if this individual of species A is growing and there are no other individuals of its species around, it has about an 80% chance of surviving. But if it happens to be growing near another individual of its species, that survival probability drops to 75. And we can compare that to species B, which also has an 80% chance of surviving if it doesn't have any neighbors of its own species. But if it is growing near a neighbor of its own species, that chance of surviving drops to 50%. So this species experiences much stronger negative effects of conspecific neighbor density. And before I go on, I want to point out a challenge of working in the tropics. When you think about working in the tropics and the challenges, you think of things like it's hot, it's buggy, there are large predators. What you don't necessarily think about are the statistical challenges. And actually, it's one of the things that turns out to be most annoying about working in the tropics. So this high diversity, which sounds amazing, also means that most of these species are very rare. And if you want to do statistical analyses, that's a problem, because even with these large data sets, many of the species, you have very small sample sizes. And so to be able to ask these kinds of questions about differences among species, you need to have lots of data. So that's good, I have lots of data. But you also need to have fairly sophisticated statistical models. So in my lab, we use things like mixed effects models or hierarchical Bayesian models that allow us to make predictions or model the dynamics of these different species, even when some of those species have quite small sample sizes. And so I'm not going to go into the details of that in this talk, but I'm happy to talk to people about it afterwards if you're interested. So using this approach, we asked, does the strength of conspecific negative density dependence vary among species within this community? And the answer was yes. And this is actually an older result that I'm showing you, not just because it's pretty cool when you get the cover of Science, and you should milk it for as long as possible, but also because everything that I'm going to talk about, or a lot of what I'm going to talk about in my work now, is an offshoot of this previous finding. So what we found, what I'm showing you here is the effect of conspecific neighbors on seedling survival, both if those neighbors are seedlings or adult trees. And this is the strength of that neighbor effect from the model. And this is a histogram showing the number of species in the community that had that strength of the effect. And the dashed line here is the zero line. So the first thing you'll notice is nearly all of the species in the community their probability of survival is lower if they're growing near individuals of their own species. So this is clear evidence supporting this idea that it is bad to be growing near neighbors of your own species. But the other thing that's even more interesting is how much variation there is in the strength of that effect. And it might not be immediately obvious, but we can compare that to how much variation we see in the impact of heterospecific neighbors, so neighbors of other species on that seedling survival. And what you'll notice here is those impacts are kind of centered around zero. There's almost no effect on seedling survival of these neighbors that aren't of its species, and there's very little variation compared to what we see up here. So we see this wide variation in the strength of density dependence among species. So next I wanted to ask, well, what is this variation related to? What could explain this? And one of the first things that we thought of was, is the variation in the strength of these Negative conspecific neighbor effects related to species abundance in the community. Are common and rare species doing something different? And we had two alternative hypotheses. The first one was that common species suffer stronger conspecific negative density dependence because they support more host-specific natural enemies. And so if you think about it, if you're a host-specific natural enemy and you're looking to specialize, you're an insect herbivore looking to specialize in a particular species, it would be, the smart thing to do would be to choose the species that you find more often, right? You wouldn't want to specialize on something that you almost never find. And so we thought these common species must be having more of these host-specific natural enemies, which would lead them to suffer more when they're growing near other individuals of their species because they'll share these enemies. But we came up with an alternative hypothesis that sort of looked at this from the other direction. And we thought, well, maybe rare species are rare because they suffer from these stronger negative effects of being near conspecific neighbors. And actually, the second hypothesis is what turned out to be correct. Rare species experience stronger negative effects of their conspecific neighbors. So this is species abundance in the 50-hectare plot on Barrow, Colorado Island. And this is the strength of that conspecific neighbor effect on seedling survival, and here's 0. So again, most of the species do worse when they're growing near individuals of their own species. But what you can see is that species that are less common in the community, they are much more sensitive to when they're growing near a conspecific neighbor. The common species barely seem to care. And if you think about it, that makes intuitive sense, because how would you get to be really, really common if your survival dropped every time you grew near an individual of your own species? And so this was a really intuitive result. But what I want to point out is this is all based on observational data, and what we really didn't know was why. What's the mechanism? And there was this very fortuitous coincidence where it turned out this other group that I know all of them, but I didn't realize this is what they were working on. They were looking at what's called negative plant soil feedback. And essentially what that means is they were looking at the impact of species-specific pathogens in the soil. And they were doing these experiments where they were looking at how sensitive species are to their specialist soil pathogens. And what they found was exactly what we found, that species that are rarer in the community are more sensitive to their host-specific soil pathogens compared to species that are more common. And this pointed to a mechanism, that it is something to do with the pathogens in the soil specializing on these species that is driving this pattern that we see in the larger community here. And so this led to a current NSF-funded collaboration that I want to tell you about. So this is Scott Mangan, who is the first author on the pathogen study I just told you about. And we've joined forces with Jim Martin and Claude DePamphlis that are genomicists from Penn State. And so, well, why do we have genomicists? Because we really want to dig deep into understanding this mechanism. So maybe rare species are more sensitive to their natural enemies, to their pathogens, but why? And so we had this hypothesis. Now, I'll walk you through it. It's the idea that species that are rare, that are locally rare, have small population sizes, and they experience stronger negative effects to their conspecific neighbors because they tend to have a lower diversity of pathogen resistance genes. So pathogen resistance genes are the genes that plants have that allow them to recognize a pathogen as something that they don't want getting into them. And what happens is if a population has a low diversity of these pathogen resistance genes, it makes them more genetically uniform, and that allows them to transmit pathogens very easily to nearby conspecifics. And the underlying assumption here is that pathogens are specializing at the genotype level. And if you have read anything about agriculture, this isn't surprising, right? You plant the same exact variety of corn and you get it wiped out by these pathogens, but if you have genetic diversity in your crop, then you have some protection. But almost no one has looked at how genotype-specific pathogens might be affecting the ecological dynamics in natural communities. And so what we think is going on is this negative feedback loop. And so if we start over here with relative abundance, so rare species have these small population sizes that result in low genetic diversity, and these are due to things like genetic bottlenecks, inbreeding, genetic drift, and so they have low resistance gene diversity. And that facilitates the spread of these pathogens among conspecifics because they're all susceptible to the same pathogens. This, in turn, means that they suffer stronger negative density dependence. And because they suffer strong pathogen-mediated density dependence, it prevents them from increasing in abundance. And so they're stuck in this loop where species are kept rare by their host-specific pathogens. And they can only break out of that loop if they gain genetic diversity through immigration coming in from other populations. And so we went to NSF with this idea and wrote this proposal, and we asked for a whole lot of money. They came back and said, well, this is really cool, and this is a really interesting idea. You have zero preliminary data. And so what they did was they said, okay, we'll give you some money. We'll give you a fraction of what you asked for so you can try and go out and show us that this might actually be occurring. So we got enough money to go out and look at 6 focal species in the BCI plot. And these are maps of those species here. And what we did was we randomly selected 5 fruiting trees from the population of each of these species, and these species vary in abundance. And from each of those trees, we collected 15 seeds, and we grew them up in the shade house down in Panama. Here's a picture of them in their nice little pots growing. And after about 2 months, we did nothing to the control group. But then we had 2 different experimental treatments where we added either live soil or salicylic acid. So live soil collected from the forest that's presumably full of their pathogens, or salicylic acid. And both of these were designed to turn on those resistance genes and express those genes. And then after about 48 hours, we ripped those poor little seedlings from the soil, washed off the dirt, froze them, and shipped them up to Penn State to our collaborators. And what they've been doing up there is isolating RNA and doing things like Illumina sequencing, ultimately to generate a metric of resistance gene diversity. And what we found was consistent with our expectations. We found that rare species have a lower diversity of resistance genes compared to more common species. So this is consistent with what we had predicted, that lower functional diversity of our genes, that locally rare species have a lower functional diversity of resistance genes, these pathogen resistance genes. But does that explain these patterns of conspecific negative density dependence? And the answer again was yes. So this is this metric of resistance gene diversity going from lower to higher genetic diversity. And what we found was that this matched up very nicely with these estimates of conspecific negative density dependence that came out of my previous study. And so this was pretty compelling preliminary support for this hypothesized feedback loop. And so we went back to NSF and said, see, look, it worked. And they said, okay, here's the rest of the money. And now we are working on tripling the sample size of this study, as well as using these genetic techniques to do other things like identify the pathogens that are actually in the roots and looking at how the microbial communities in the different root systems are similar for different seedlings from different species. So that's one project we have going on in the lab right now. At this point, we're pretty much done the on-the-ground part in Panama, and all the samples have been shipped over to Penn State where they're doing the genetic lab work. But in the meantime, back at Yale, we've been thinking about, well, what else might contribute to this variation among species in these negative conspecific neighbor effects? So this is just the graph from before showing this wide variation. And we know that some of it is explained by abundance, but not all of it. And so we got to thinking, well, what else might explain this? And the first thing that came to mind was species life history strategies. And what I mean by this specifically is shade tolerance. Tropical tree species vary widely in their shade tolerance. Some species are able to germinate and establish in the shade and grow very, very slowly, while other species absolutely cannot make it in the shade. They require these really large high light gaps to regenerate. And this shade tolerance is governed by a trade-off, a physiological trade-off between being able to grow fast in high light and being able to survive well in the shade. And this is a well-documented trade-off in tropical trees. And we hypothesized that this variation in life history strategy might be related to the strength of these conspecific neighbor effects, and that a species location along this growth mortality trade-off axis might influence the degree to which it's impacted by conspecific negative density dependence. And we thought this because species that are slower growing and shade tolerant tend to have traits like high wood density and better defended leaves that would leave them less susceptible to pathogen or insect attack. And so if you're less susceptible to pathogen or insect attack, you should be less sensitive to conspecific neighbor density. And so a former postdoc in my lab, Dr. Yan Zhu, who was here for a couple years, she's now an assistant professor at the Chinese Academy of Sciences in Beijing. She led this study that came out earlier this year in Ecology Letters, where we looked at whether life history strategy explains density-dependent survival. And we did this not just for seedlings, but we used all life stages from seedling, sapling, juvenile, and adult trees to test for this relationship. And what we found specifically at the sapling and adult stages was consistent with our hypothesis. Faster growing species were more sensitive to conspecific neighbor density than slower growing species. So the x-axis here is conspecific neighbor density. So having no neighbors of your own species to an increasing density. And this is the predicted probability of survival from the model. And what you can see for fast growing species, survival drops very rapidly as conspecific density increases. But for these slower growing species, it still drops, but not anywhere near as steeply. And so we had support for this idea. So far, what we know is that there's wide variation among species in their sensitivity to these conspecific neighbor effects. It's related to species abundance. It's also related to species life history strategy. But okay, so what? The problem is when you just look, even with the kind of long-term data we have here over more than a decade, it's really just a small window into the lifetime of a tropical tree. These trees live for potentially decades or hundreds of years. And if we want to know what the implications of this are for the community over relevant time scales, so the scale of multiple generations, we can't actually do it using observational data alone. And I was lucky enough about a year and a half ago to recruit Simon Stump as a postdoctoral researcher in my lab. And Simon is a theoretical ecologist, and his former work had focused on constructing theoretical models, so using computer simulations to test species coexistence mechanisms. And so he came to the lab and we started thinking about what would be the most exciting thing to work on. And what we decided is it would be interesting to look at how this variation among species in their sensitivity to conspecific neighbors, what are the implications of that for species diversity and community stability over multiple generations? And so Simon built this model, this computer simulation, and this is his lovely drawing of what it actually does. So you have this, in this case, 3 different species. Each color is a different species, and they produce seeds, and some of those seeds are dispersed away. And then he simulates that some of those seeds die from conspecific negative density dependence. So the yellow seedlings are more likely to die under a yellow tree compared to red or blue. And same with blue and red. And what he did was ran models where the negative effect of being underneath your own species was either the same for all species or that strength of that effect varied among species. And then he simulated an adult tree dying and that tree's spot being taken over by one of those seedlings that survived. And so he ran these simulations, and what he found, or what we found, is that variation in conspecific negative density dependence actually makes communities less stable. And this is pretty controversial because everyone always thinks about conspecific negative density dependence as this mechanism that maintains diversity. This is a diversity maintaining mechanism. But what we found is that if you allow that strength of that effect to vary among species, it reduces the number of species that can coexist. So this is a graph from our paper that came out recently in Ecology Letters, and what we found was that, so if you allow all species, this is a community that he started with 15 species, and if we allowed all of them to have the same exact strength of density dependence, all 15 species were maintained. But if you kept the mean strength of density dependence the same, but allowed it to vary among species, very quickly you started to lose species from the system. These different lines here actually correspond to different degrees of seed dispersal away from parent trees. It turns out that seed dispersal is extremely important for these interactions. I won't talk about it too much today, but that was another finding that came out of this. And he parameterized this theoretical model using estimates from my site in Panama. And what we found was that in some cases, this variation, when there's variation in conspecific negative density dependence, it actually decreases rather than increases the tree diversity you expect to find in a site. And as I said before, this is controversial because every time anyone finds evidence of conspecific negative density dependence, they say, and therefore, this maintains diversity in tropical forests. This is evidence that you have, that this is a mechanism maintaining diversity. And what we showed is that's not always the case. This diversity maintaining mechanism can sometimes actually cause decreases in diversity. So I've just taken you on a really deep dive into the key ecological mechanisms that maintain diversity in tropical tree communities. And I just wanted to end by talking a bit about how we've been looking at whether anthropogenic disturbances alter these key interactions. And in particular, I want to highlight work that was led by my PhD student, Meghna Krishnadis, who defended her dissertation earlier this year and will be officially graduating in December. And she was working in the Western Ghats biodiversity hotspot in southern India here. And she worked on this is actually a tea plantation. These fields here are tea. But within this landscape, you have all of these fragments, these forest fragments remaining. And when you have fragmented forest, you get something that's called edge effects. Edge effects are areas that are close to forest edges tend to be hotter, they tend to be drier, they have higher light. And these edge effects can persist, depending on the specific effect, anywhere from a few meters up to hundreds of meters as you move from the edge into the forest interior. And most people have thought about, well, how these changes in light and moisture and temperature affect plants, but nobody has really been looking at whether these edge effects alter the ecological interactions that maintain tree species diversity. And so Meghna's project set out to test just that. What she did was across this landscape at multiple sites, replicated across this landscape, she set up sites at increasing distances from the forest edge, moving further and further away from that edge. So at these points, you expect to see these strong edge effects and then much less as you move. These sites were about 90 to 100 meters away from the forest edge. And she monitored seed arrival into these sites as well as seedling recruitment. And what she found was that for seeds, this is showing the distance into the forest from the edge, and these are sites that are right at the edge, 20 to 30 meters, 50 to 60, 90 to 100, and this is a metric of diversity. For seeds, there was no significant difference among these sites in the diversity of the seeds that were getting there. And if anything, it was a little bit lower at the furthest distances from the edge. But when she looked at the diversity of seedlings coming up at these sites, what she found was there was this drop in diversity at sites less than 60 meters from the edge, but that diversity was maintained and was significantly higher at sites 90 to 100 meters in. And this suggests that something is happening here. We're losing some kind of diversifying process. And so she designed this experiment to look at what we are losing. And of course, because in our lab we think a lot about pathogens and insects and their role, she set up this experiment where this is showing again edge to interior. These are plots 0 to 5, 20 to 30, 50 to 60, and 90 to 100. And the black circles here are control plots that she didn't do anything to. And then she had plots where she sprayed either insecticide to remove insect herbivores or fungicide to remove fungal pathogens, or both. And so if we look at sites right at the edge and within 60 meters, applying insecticide and fungicide had no effect on seedling diversity. But if we move to this site where in the control plots we saw higher seedling diversity, when you spray those plots with insecticide or fungicide, when you take those insect herbivores and fungal pathogens out of the community, you see a drop in diversity. And what this tells us is that this higher diversity of seedlings away from edges is because you have fungal pathogens and insect herbivores that promote diversity, and that we've actually lost these diversifying interactions at sites that were less than 60 meters from the edge. And the implication of this is actually massive. If we think about the global magnitude of forest fragmentation, there was a nice review study out a couple years ago in Science Advances where they looked at forest fragments globally. And what I want to draw your attention to here is this figure, where they looked at the proportion of remaining forest in the world that is within different distances to the edge. And their main conclusion was that 70% of remaining forest is within a kilometer of the edge. But what I want to point out even more, emphasize even more, is 20% of remaining forest globally is within 100 meters of an edge. And what Meghna has shown that if you are less than that, you have lost these diversifying interactions that maintain diversity. And so the implications of that globally could be massive. And then finally, I want to end on what might be a happier note, but it's still too soon to say. I want to tell you about some work that is just starting about whether these diversifying interactions can be recovered in tropical forests over time. And these days, in terms of tropical forest conservation, it's pretty much gloom and doom. But one of the bright spots is the fact that you are getting massive areas of regeneration, of forest regenerating, often on abandoned agricultural lands. And so you are seeing forest coming back. And certain aspects of the forest structure, forest biomass, recover pretty quickly within a few decades. But what we still don't know is, are these ecological interactions being recovered in these systems? And it's really my hope that they are, but we don't know that yet. And so I'm just starting a new collaboration with Dr. Daisy Dent. She's a professor at the University of Stirling in the UK, who's also been working in Panama, and she has this nice, what's called a chronosequence. She has forest plots in which she's been monitoring adult trees along with her collaborators at sites of forests that are differing ages, going from about 5 to 10 years old up to 80 to 100-year-old forest, as well as old growth forest in this landscape. And we're interested in looking at these patterns of conspecific negative density dependence, as well as plant pathogen interactions more generally in these regenerating secondary tropical forests. And we're interested in comparing both secondary forests to old growth, but because we have this nice chronosequence in place, we can also look at how these interactions change over time from younger to older forest. And so what we're interested in is whether these interactions are lost in the first place, how quickly they're recovering. And another idea that's really exciting is actually that plant pathogen interactions might play a role in driving succession. So if you think about it, as a species gets to a site, it starts to increase in abundance, you expect its host-specific natural enemies to build up as well. And so when the next species manages to disperse seeds to that site, it's going to have an advantage because it's escaped its natural enemies. And so these negative interactions with insect herbivores and plant pathogens might actually facilitate secondary succession in these landscapes. And that's one idea we're interested in testing. And so I'm really hopeful that this might actually be a happy story, but unfortunately, we're going to have to wait a few years till we know the answer. But with that, I'd just like to acknowledge my collaborators on all of these different projects, in particular my field crew down in Panama, who are truly amazing. I'd also like to thank the graduate students and postdocs in my lab. They are a really supportive and productive and fun group that I really enjoy working with. And finally, sources of financial and logistical support. And I wanted to especially thank the staff here at FES, from the grants office to the dean's office to facilities to IT to communications. So much of what I do is possible because of what you do. And so speaking here in FES, I wanted to really make sure to thank you all for that. So with that, I'll thank you for your attention, and I'm happy to take any questions.
So in BCI, where you found these effects, and there's hundreds of people researching, do you think that there's some negative effect of people trudging dirt around on their boots from rare tree to rare tree and moving pathogens around? Has there been a change over time as people have been there that you've noticed?
That's a great question. And it's not the question I first thought you were answering, which is just, are these little seedlings getting trampled, which is something we actually looked at. We looked at whether there was a sort of researcher impact at this site on the seedling dynamics, and we didn't find evidence of that. We published a study on that. The idea of spreading these pathogens around is a really interesting one. I don't think anyone knows the answer for plants. What we do know in Panama, in particular, you hear a lot about this chytrid fungus that's been killing off amphibians. And I know amphibian researchers are very careful when they go to a site. After they leave, they take their boots off and they bleach them and they clean them really well before they go to the next site to make sure they're not spreading these pathogens. Man, that's a really interesting question now that you mention it. And I might start cleaning my boots more.
I have just another site-related one, so I'm inserting it here. I noticed you didn't mention the question of introduced species versus native species, and that ties into the land use history going back maybe before the island. So maybe just elaborate on that a little. And also, you talked very specifically about host-specific pathogens. I wonder if you could say a little bit about more generalist pathogens, because that would begin to interfere with your theory here if there were pathogens that were, or insects that were more able to attack the species other than just a specific species.
Okay.
Thank you for your nice talk, by the way. Thanks.
Yeah, thank you for your nice question. So for the first question about invasive species, in terms of invasive plant species, there's actually very few that we find in the 50-hectare plot. In tropical forests, with some exceptions, invasive species aren't as much of a problem. Some of those exceptions are often tropical islands, like in Puerto Rico and Guam and Hawaii. In other sites, invasive plant species tend to be more disturbance adapted and take advantage of disturbance in forests and higher light conditions. In Panama, there's a particularly nasty invasive grass that a former student of Mark Ashton's had worked on, this Saccharum spontaneum, that if you cut the forest, it just takes over and the forest can't come back. And seeds of those actually arrive into seed traps on BCI, but they never germinate in the shade. So that's not such a problem here. Along this chronosequence of secondary forest, I think once the forest comes back, you stop seeing a problem with those species. The problem is if they get in first, they often prevent succession from happening. And then your second question was about generalist pathogens. And so I think generalist pathogens don't necessarily negate this. You can have, and I'm sure you do have, both generalist pathogens and specialist pathogens. And what we know from pathogen host range studies is typically there's a phylogenetic signal, meaning that a particular pathogen or insect usually doesn't only attack a single species, but it usually attacks a suite of species that are of the same genus or of the same family. What matters for these dynamics is not whether it's truly host-specific, that's the only thing it eats, but whether it has a stronger impact on one species versus other species. And there is a nice theoretical study, again, using similar kinds of simulation models to look at if you relax that assumption of host specificity and allow there to be this phylogenetic signal, do you still get diversity maintained? And they found that you do.
Thanks. So I'm wondering about your seed dispersers that you put up that great slide about. It sounds like you guys have got some work going on about that, and I'm wondering, A, what kind of things you're looking at, and B, if you think there might be some kind of trade-off within these plants that they either put their energy into defending themselves against their pathogens or attracting really good seed dispersers.
So in terms of seed dispersers. We do, especially for Barrow, Colorado, because it's been studied for so long, we have a really good idea of, at the very least, if species are wind dispersed or animals dispersed. The majority of species are animal dispersed by birds or mammals or even bats, and there's a lot of secondary dispersal on the ground of rodents moving things around and caching them. In terms of a trade-off between defending yourself against pathogens and selecting for dispersal, I'm not sure. I'd have to think about that a little bit more. What we do know is that the shade-tolerant species that tend to be less susceptible to pathogens produce larger seeds that tend to be animal dispersed. And the pioneer species, the light-demanding species that are more susceptible, are wind dispersed. And so, yeah, so looking at the implications of that for density across the landscape versus how well defended things are is definitely an interesting question. I don't think it's an obvious answer. I don't think there's a clear answer to that. One study that we recently did that was really interesting, a postdoc in my lab worked on this one bat dispersed species and found actually that there was no effective negative density dependence, that they did find growing at higher conspecific densities. And this is a bat dispersed species in a bat that has roosts, and they drop the seeds at their feeding roosts, and so you get these high conspecific densities away from the parent tree. And so seedlings did better if they were away from a conspecific adult, but they actually did better at high conspecific densities because they corresponded to these bat roosts. And we think that's because the bats also defecate underneath the tree, and that makes it really nutrient rich, and so it's this happy place for seedlings. And so I think when you bring dispersers into it, it gets really interesting, but it also gets really messy. And so we definitely need to do more work on that, though. Okay. That's okay. Go ahead and then.
Very curious to know about these slower growing species, whether or not their lower rate of infection or predation is just due to the fact that their photosynthetic rate, they produce photosynthetes at such a lower rate that they're less attractive, and it's one way to evade attack or predation.
Yeah, that's definitely a possibility. But what we also know about these species is they tend to have better defended, tougher leaves. And so this kind of whole life history strategy seems to be about being able to grow fast versus being able to survive for a long time in the shade. And they make these leaves that actually live much longer. The individual leaves live much longer than the faster growing species, because if they lose some leaf tissue to herbivores or pathogens, they really don't have the resources to replace it. And so when they do invest in leaves, they also invest a lot into defending those leaves against attack. I think.
I was going to say, we just have just a few more minutes, so maybe we'll hear, there are three, or actually a bunch of hands up. Maybe we'll just hear a bunch of questions, and then you can pick and choose depending on.
Okay.
Okay, thanks. I'll go very quickly. I was curious about all the trouble that you were talking about fungi creating for the survival of these seedlings, and in Magna's study showing that when you have when you use fungicide, that things actually do worse, I think. And I was relating that in my mind to Suzanne Simard's work on looking at mycorrhizal communities and how that actually promotes like species potentially. And I just didn't know if you had any thoughts about how that applied in perhaps this tropical context.
Yeah, so, okay. Or do you want to, okay, you want me to take, I'm going to take all the questions and then.
Just one comment. Did you ever check these species for their response in their central niche versus their marginal niche? It can't just be photosynthesis, as someone was talking, but that's a problem because it works for muskrats as well as plants.
Okay.
And so